Good morning, everybody. Welcome to this postdoc talk of the student network of extracellular vesicles. Today's presenter is Carolina Balbi from the Laboratory of Translational Research, Cellular and Molecular Cardiology in Bellinzona, Switzerland. And she will talk today about her work about EVs in coronavirus patients. We learned a lot about this virus in the past couple of years. The pandemic is almost over, but you never know what else is going to come. So I want to hear all about it. Please, Carolina, the floor is yours.
Thank you. I'm going to share the screen. Okay. So are you able to see my screen in the presentation mode?
Yes, it's good now.
Okay. So good morning, good evening, or good night, everyone. I'm Carolina, and I'm a postdoctoral researcher working in laboratories for translational research. And I'm super happy, I have to be honest, to present in the SNEV PostDoc talk because I'm part of SNEV, and I really like and enjoy being part of SNEV, and I am proud of the mission of SNEV to connecting young researcher and early career research, PhD student in a topic, in such an interesting topic as extracellular vesicles. I put 2-3 slides just to present a little bit myself and my story with extracellular vesicles. So I started my career in research with my master degree at the University of Genoa in Italy. After my master degree, I moved with a fellowship in Paris, in France, where I studied the long non-coding RNA during the developmental process. Then I started my PhD back in Genova in translational medicine, and during my PhD I moved for a period in the Leiden Medical Center in the Netherlands. At the end, in 2018, I started my postdoctoral fellowship at the Istituto Cardiocentro Ticino in Bellinzona in Switzerland. So my story with extracellular vesicles started during my PhD, where my project was aimed to understand the role of a secretome from a subset of stem cells, the human amniotic fluid stem cells, as cardioprotective, in a myocardial infarction model, as cardioprotective activity of the secretome. So we applied secretome after myocardial infarction injury, and we found that this secretome was able to decrease the infarct size to cardioprotect the cardiomyocytes and also to induce cardiac regeneration. As you know, secretome is composed by soluble proteins and extracellular vesicles. So we decided to dissect which one was the main player of such cardioprotection. So we perform an experiment using only the soluble proteins or only the extracellular vesicle from the secretome. And we found that the extracellular vesicles were able to recapitulate almost all the beneficial effect of the secretome, the total secretome. So finished my PhD, I decided that I wanted to contribute in Bellinzona at Cardiocentro Ticino, where I continued to study the therapeutic effect of extracellular vesicles in myocardial infarction and also in a cardiac toxicity model. As many of you interested in extracellular vesicles, when there was the COVID-19 outbreak, I became interested in which role has extracellular vesicles in COVID-19 infection. So you know that SARS-CoV-2 is able to bind endothelial cells to the spike protein on its surface. The binding of the spike protein is with the ACE2 receptor, and this binding leads to endothelial damage, and the endothelial damage induces cytokine storm and inflammation. It's important to note that inflammation and extracellular vesicles are tremendously related. So I reported only 2 examples here, but there are thousands in the literature of inflammation stimuli and extracellular vesicles. In the first paper, you can see that the authors treated the in vitro microglial cells with LPS, and then characterized the vesicles compared to the control microglial cells, not PBS treated. And they found that treatment with LPS induced an increase of vesicles, size of the secreted vesicles, but not only a change in the morphology, there was also a change in the cargo. So vesicles from LPS treated cells were enriched in pro-inflammatory and pro-apoptotic proteins such as iNOS or procaspase-1. In the other side, there is another paper. This one was done in another lab of Cardiocentro Ticino, and they perform an analysis in vivo on plasma extracellular vesicles in a rat model of myocardial infarction. So they analyzed the vesicles before the myocardial infarction and after the myocardial infarction surgery, and they found that after myocardial infarction, in the acute phase of inflammation, there is an increase in the secretion of the vesicles, so a higher number of vesicles, but also that the cargo of these vesicles was different. So vesicles in the acute phase of inflammation are more enriched in iNOS and CD68 protein, so pro-inflammatory protein. For all that reason, we decided to investigate the circulating extracellular vesicle from SARS-CoV-2 patient. This work was done thanks to the help of Sara and Jacopo, and thanks also to our collaboration with the hospital, because we are a lab of the hospital, so we had the possibility, or at least is easier for us to get patient samples. So we took the blood samples from healthy subjects and from subjects that were symptomatic and underwent to nasopharyngeal swab. And after the result of the swab, we divided these 2 samples in COVID positive and COVID negative. This is important to underline because our COVID negative samples were samples from patients that were symptomatic, so patient with inflammation, patient with influenza, but not influenza due to COVID. After the receiving of the samples, we performed serum separation, and then we started some low-speed centrifugation to get rid of cell debris and larger vesicles. And then this pre-cleaned serum was incubated with fluorescent beads with 37 different antigens on the surface. And a day later, these beads were visualized by flow cytometry thanks to antibody against CD9, CD63, and CD81 in order to visualize the extracellular vesicles. So first data that we found was that extracellular vesicles from COVID positive patient were higher in number compared to the COVID negative and the healthy control. So as you can see here, the higher in number is measured as increasing mean fluorescent intensity of CD9, CD63, and CD81 that are tetraspanin present on the vesicle. So higher is the intensity, higher the number of tetraspanin recognized by the antibodies. And as you can see here, the amount of the COVID negative vesicles is higher than the one from the healthy controls, underlying that this patients were patients with influenza again, so with inflammation. Inflammation is something related to the increase of the vesicles. Then we analyze the other 37 surface markers. And as you can see here, we found a cluster of 7 surface markers that were enriched in extracellular vesicles from COVID-positive patient, but not from COVID-negative and not from healthy subject. We took these 7 markers and we performed a correction for age, sex, BMI, and pneumonia. After the correction, we found that these markers were significantly increased in extracellular vesicles from COVID-positive patient compared to the negative and the healthy control. Among these 7 markers, there is one that is particularly interesting, that is CD142. CD142 was the one with the higher diagnostic performance, with also an area under the curve of 0.830 and a sensitivity of 79.4 and a specificity of 81.8. But CD142 was not only interesting because of his property as diagnostic markers. It's also interesting because CD142 is better known as tissue factor. So this is a model proposed by other studies of the role of SARS-CoV-2, of the disease cellular role of SARS-CoV-2. So SARS-CoV-2 is able, as mentioned before, to bind AC2 to induce endothelial damage. But the endothelial damage leads to inflammation and also activation of a tissue factor. And we know that one of the major complications of COVID-19 positive patient was related to thrombotic events. So tissue factor and COVID are something strictly related, and the things that we found tissue factor on the surface of the vesicles could derive from this activation of endothelial cells, thanks to the binding of the virus. So we decided to investigate more the role of tissue factor on the surface of the vesicles. And firstly, we analyzed again only the vesicles from the pneumonia patient, because we had sample of COVID-negative and COVID-positive patient with pneumonia. And again, we found that in COVID-positive pneumonia patient, the number of the vesicles was higher. And again, among the 7 markers visualized before, 5 remain still significant, and still CD142, so the tissue factor was the one with the higher diagnostic performance. So we isolated the vesicles in order to visualize tissue factor, and we performed Western blot. As you can see here, we performed Western blot for some extracellular vesicle-specific markers such as CD81, synthenin-1, TSG101, and also we perform Western blot for contaminants to see a decrease of contaminants in our preparation, and contaminants in serum are mainly lipoproteins, so we perform Western blot for ApoB48 and ApoA1. And then Western blot for CD142 and tissue factor that confirm the presence of this protein on the vesicles. As you can see here, enriched in COVID positive, visualized by Western blot, but we also perform activity assay of this tissue factor, because tissue factor is not only important that is present on the vesicle, but is important that is active, so that can start the can exert his procoagulation activity. As you can see here, tissue factor was more active in extracellular vesicle. Here, the assay was done on the same number of vesicles, so it's not related to the more vesicles present in the sample. So we isolated the vesicles, we measure the vesicles by NTA, and then we perform CD142 activity assay, and we found that for the same number of vesicles we have more active tissue factor on COVID positive patient, and this was also true for a patient with pneumonia from both negative and positive COVID-19. Coming back to this slide, with this model, is known from the literature that one of the cytokine more expressed in COVID patients is TNF-alpha. So we decided to measure TNF-alpha in our samples, and we found that there is also a direct correlation between TNF-alpha and tissue factor expression of the vesicles. Furthermore, we mentioned that SARS-CoV-2 is able to bind endothelial cells and induce endothelial damage. So we analyze we decided to analyze from in the subset of the markers on the surface of our endothelial, our extracellular circulating extracellular vesicles, which are the from which kind of cells these vesicles are obtained. And we found that in COVID positive patients, there is a significant increase of vesicles derived from the endothelial cells compared to COVID negative, but mostly compared to healthy control. So suggesting that the endothelial damage caused by the virus is really, really important to induce an increase in the secretion of the vesicles and an increase in the secretion of tissue factor expressing vesicles. So this is just a slide to show what we propose. We propose that after the infection, there is an increase in tissue factor in TNF-alpha amount in the circulation, and this TNF-alpha induce activation of endothelial cells, damage of endothelial cells, and release of extracellular vesicles with active TNF tissue factor on his surface. And it's important that tissue factor is expressed on the vesicles, because vesicles may exert a procoagulant activity in different districts in the body, not only in the district with the damage. That is really something to take into account. So I've just put together some slides from other papers just to underline really the importance of the study of circulating extracellular vesicles. So here, this is a super nice study, in my opinion, that was done again on circulating extracellular vesicles from COVID patients, and they took the vesicles from asymptomatic to severe patients, so at different stage of the disease. And the authors here isolated large and small extracellular vesicles, and they compare large and small extracellular vesicles in vitro, treating endothelial cells, and they found that these vesicles were able to induce cytotoxicity in endothelial cells. So they are not only important as mediator of activity, but they also are important to increase the damage of the cells. Both large and small extracellular vesicles induce endothelial damage, and then they perform a proteomic analysis of these vesicles, and they found that one of the most enriched protein is again tissue factor. So tissue factor is enriched in all the stages of the disease, especially in the severe one. Interesting, they also made some correlation, and they found that the activity of tissue factor is related to the length of the hospitalization and to the disease severity, analyzing both large and small extracellular vesicles. So really a tissue factor of the extracellular vesicles is important, is related to COVID-19 and has an effect on the disease. This is another paper that went out early this year, super interesting, because they again took the vesicle from mild and from severe COVID-19 patient. They isolate the vesicle and treat the T cells, and they found that only vesicles from mild patients were able to properly activate T cells, so to properly activate the immune response. So they performed then proteomic analysis on these vesicles, and they found that extracellular vesicles from mild patient were enriched in protein that have a role in the proper activation of the immune system, while protein inside the vesicles from the severe patient are more related to a chronic inflammation. So patient with a higher disease, with a severe disease, has circulating extracellular vesicles that are enable to activate the immune system, but only to induce chronic inflammation. So they are able to increase the damage of the disease. But it's not only something related to the acute phase of the disease, it's also something related to the long term of the disease. It's now it's now known that there are some problems also months after the COVID-19 infection. Is called the long-last COVID. And in this paper, the authors decided to perform proteomic analysis on the vesicles 3 months after the recovery. And again, they took vesicles from recovered asymptomatic, recovered mild, and recovered severe patient. And what they found was that still 3 months after the recovery, there are some proteins that are up or down regulated inside of these vesicles. As example, TGF-beta1 is significantly increased in the vesicle from COVID patient compared to the healthy subject. And this significant increase in all the vesicles from COVID patient, not only in the severe, but also in the mild and in the asymptomatic one. But there are also proteins that are related only to the severity of the disease. As example, phospholipid transfer protein is enriched in a recovered severe patient and not in the asymptomatic patient. But what is really interesting is that 3 months after the recovery, these proteins are still alterated. So the vesicles has a role not only in the acute phase, but also after months after the recovery of the disease. So here I draw these slides just to underline the importance of the study of circulating extracellular vesicles, because from the bloodstream we can take extracellular vesicles derived from different type of cells. We can, for example, in case of COVID, we can took extracellular vesicles derived from immunocells, so lymphocyte, macrophage, neutrophils, and also we can isolate vesicles derived from infected cells, for example, the endothelial cells. And the study of these vesicles can lead to new biomarkers, so new markers that are important and are strictly related with, for example, the severity of a disease, or to understand to new therapeutic target, because we can discover a mechanism of action of these vesicles or the propagation of a disease, and then we can use this knowledge to treat the patient. So it's really, really interesting, not only on the COVID infection, but it's really interesting in different type of a disease. The study of circulating extracellular vesicles is a powerful tool. Okay, so with this slide, I would like to thank the lab, in particular the Professor Vassalli and Professor Barile lab, and of course you for the attention, and obviously SNEV for the opportunity. So thank you so much.
Great talk. Thank you, Carolina.
Thank you.
I think we have a lot to unpack here. So let's see if there are any questions from the audience. I didn't see any in the chat, but if you want to ask a question, please unmute yourself and also turn on your camera so we can see you.
So don't be shy.
Okay, maybe people's heads are still spinning, but I can kick off with a couple of questions. Really nice work, and I know this assay I think that you used, is that the Max-Plex assay?
Yeah.
That's a very nice way to capture EVs and do a broad surface protein profiling. At some point you showed a breakdown of what the progenitor cells were of the EVs that were circulating, and you said that in the COVID patients more EVs were derived from endothelial cells. How did you derive that? Because that's always a big question, right? You see more EVs, where do they come from? And especially in complex fluids like plasma. So can you tell a little bit more about how you derived that?
Yeah, sure. So two things. First one is that we use the MaxPlex, but we adapt a little bit the MaxPlex kit to use it as a diagnostic tool, let's say. So we didn't isolate the vesicles. We put directly the beads from the MaxPlex in the serum. That is because we wanted to have some fast screening, let's say. So adding an isolation step is going to be slower. It's going to slow the results, let's say. Then after the analysis of the MaxPlex, we perform some literature analysis, so taking each antigens that is present in the MaxPlex, and we decided we used the literature to say, okay, this is a marker of expressing lymphocyte, this is a protein expressing endothelial cell, etc. And then we clusterize these markers as cellular origin derivation. So we say, okay, if we have a higher number of vesicles with this marker, these vesicles are derived from endothelial cells. So it's a little bit empiric, let's say, but it's an indication of which cell types produce the vesicles. And of course, there are some markers that are both endothelial cell or other cell type, but it's a good indication of the derivation of the vesicles.
Yeah, that makes sense. So did you use single markers per cell type, or did you make
No, we clusterize. We clusterize the markers, yeah.
Okay, that makes sense. I had another question because you found that there was there were more EVs containing tissue factor, but tissue factor is also occurring as a soluble protein in the bloodstream. So do you think these differ in functionality, or what would tissue factor on EVs do differently than the soluble version? I don't know a lot about coagulation, but
yeah,
maybe you can,
what we didn't do was a comparison of the because the activity test was done on isolated extracellular vesicles. So we isolate them with ultracentrifugation, and then we perform the test. What we didn't do was a comparison of the, for example, the sovranatant of the ultracentrifugation and the vesicles. That is something that is going to be important to discriminate the soluble tissue factor and the tissue factor on the surface of the vesicles. But from the last SNEV postdoc talk, Oscar performed some studies showing the role of free proteins and protein binding on the surface of the vesicles, and really demonstrated that protein on the surface of the vesicles are more active than free protein, because they can bring together the receptors, so they can clusterize better thanks to the morphology of the vesicle, let's say. So I would like to say that probably is more active, the one on the extracellular vesicle, but it's something that we didn't confirm, but it's going to be interesting. There are a lot of papers now on extracellular vesicles, tissue factor expression and everything, but no one compared the soluble one and the one binding on the vesicles. So I think it's something interesting that is worth to do for sure. Thanks, Tom.
Nice. So that's on your to-do list then.
Exactly.
Nice.
Exactly.
But it really makes sense that these EVs may contribute to coagulation, because I heard something about microthrombi, so small blood clots that are not visible with regular methods that can even travel up to the brain and may be involved in long COVID. So your results really underline that. So
yeah,
that's
sure.
nice.
And the correlation really between the expression of this tissue factor and the severity of the disease is really strong. So they really have a role. It's important to study this field, in my opinion.
So what do you what do you think? Would you advise using anticoagulant therapies in corona patients, or is
it's a really good question, because they already tried to use anticoagulant, but I know that it's not only that was something that is was a little bit started, then interrupted, then started again. So they try different therapies. But I think that if you have a tool that suggests you that this patient is more prone to develop some disease, for example, they have an higher amount of tissue factor extracellular vesicles in their circulating bloodstream, this patient could be interesting to be treated with anticoagulant. If a patient don't have these extracellular vesicles, why treat this patient with anticoagulant? So having a photo, let's say a photography of the situation of a patient before the treatment using extracellular vesicles is something that it could be powerful. As I said before, our samples arrived before the swab test, so this patient were really at the beginning of the disease. So already at the beginning of a disease, you have an idea of how long is going to be his disease, how severe is going to be his disease, only on the expression of the tissue factor. So this is important to underline, because you could have an idea of how to treat your patient a little bit better than without this test, let's say. So,
nice. Yeah. So if you think about these biomarkers that are going to be used in the clinic, of course, as a doctor, you would like to have an answer as quickly as possible. So what do you think? How quickly could you test for this on EV specifically, or maybe on whole plasma?
Yeah, I think that if you want really something really fast, using an isolation method is not going to work for sure. But for example, if you bind your vesicles with beads for CD9, CD63, you have a good balance of purity and time. Let's say in this case we perform an overnight incubation, and the day later we perform the fax analysis. So it's 2 days to have an answer, but you can also shorten the time, because overnight incubation can be done a little bit in a shorter time with higher temperature. So I think that flow cytometry and beads are the only way to have a good diagnostic tool with extracellular vesicle, or some microfluidics method, but we are still quite far for having microfluidics method, in my opinion, working for the extracellular vesicles.
Yeah, you're right. That's very early in development, but who knows?
Exactly.
But these timelines, I think they're reasonable for a disease like coronavirus. I worked a little bit on sepsis, and there you want to have really a quick outcome, because these patients, they may go from okay to bad in a couple of hours. So I think my biomarker wouldn't be that good, but this sounds very promising.
Yeah, and I think also that, we are focused on extracellular vesicles, but if you find something that is not really on an extracellular vesicle, let's say on a small extracellular vesicle, but you can co-precipitate or is a contaminant of your sample, but it's still a biomarker, that's the most important things, I think. So
yeah, that's true. As long as it gives you insight in the patient, it doesn't have to be on an EV per se.
Exactly.
I totally agree. But if you claim that it is, then you should show it.
Yeah, it's true.
Nice. Okay, we had some two-way traffic. We have a question from Maimona in the chat. Do you want to unmute yourself?
Okay. So I'm gonna read the question. If I'm able to expand the chat. Okay, can you talk about the obstacles you faced during doing the project and how you could overcome them? So, thanks for the question. You mean there are different type of obstacles. First of all, performing this work during a pandemic. So everything was blocked, then everything was closed, you know that. But our lab is a lab from the hospital, so we were under the hospital rules, let's say. So we never stopped. We continued to go to work. We, of course, put some rules, internal rules to between us, so we decided to not stay more than 2 persons in a room, something like that, but at least we can continue to work. And this was really important because we got the fresh samples from the patient that underwent to the hospital to perform the screening. So this is something that for sure was an obstacle, but we overcome quite good because of the help of the hospital. Secondly, another obstacle was that in the beginning we didn't know how to use the serum, because we didn't know if the serum was infected, if we can use the serum and all this stuff. So we performed some, together with the hospital, some analysis of PCR in the serum, and we found that the viral amount on the serum is completely zero, so there is no transmission of the virus through the serum, or at least the amount of the detected particles, virus particles in the serum is really, really low. So we were quite safe using these samples. I don't know if there are some other specific obstacles that you have in mind, please ask, but I think there are no others at least in my mind. That was the 2 obstacles that we faced and how we overcome that. So I hope I answer. Okay, methodological obstacles. Okay, not really, because we used the MaxPlex really for a lot of works, because we performed and the one really expert on MaxPlex is Jacopo, the guy that performed the work with me. He's a medical doctor, and he's really interested in biomarkers and all this stuff as working in the hospital, and he performed MaxPlex on serum on different type of disease, and he together with Sarah make a good protocol on this. So when we had the sample of COVID, we already have a protocol set up, and we were quite fast. But in the beginning, they spent quite a lot of time to see if MaxPlex could work only in this directly on the serum or directly in the plasma without the isolation of the vesicles. So they compared isolated vesicles with not isolated vesicles, and they find a good balance to have a diagnostic tool, or at least a tool that could be used in a diagnostic way quite fast without the isolation of the vesicles, with good results without any or at least without not so much interference of the protein present in the serum that are not get treated without the isolation method. Don't worry, it's not a problem. So yeah, that was the only methodological obstacles. But as I mentioned, when we started this project, we already had the protocol set and worked quite good. But MaxPlex is really a good kit, I have to say. It's worked quite well, and it's really informative for circulating extracellular vesicles.
Exactly, and because of the beads, you can use, I would say, any kind of flow cytometer.
Yeah, exactly. We use a normal flow cytometry, is Cytoflex. We have no special settings and everything, so it's really easy.
Super. Are there any more questions? No. Oh, yes.
So is PCR on both. So we perform, the question is PCR performed to check the presence of a virus. Was it done on whole serum sample or final EV sample? On both. We perform a digital PCR to be really more accurate, let's say, and we couldn't find the presence of the RNA of a virus on both isolated extracellular vesicles or in the whole serum. So with the volume that we used for the screening, because I have to say that MaxPlex was done on 100 microliter serum, so really not in a larger volume of serum. You can work with really 100 microliters and no more of samples. So, thank you for the question. Thank you, Vera. Okay.
Great. Well, if there are no further questions, I think we can wrap up.
Yeah.
Thanks, Carolina, for the interesting talk and answering all of our questions. Okay.
Don't worry, Vera. Absolutely. If you need something, you can also write me.
Yeah. Sorry.
Okay.
I have one more question, if possible.
Yeah, go ahead, please do so.
Yeah. There are some rare cases where vaccine recipients developed some clot formation, right? And they had some kind of stroke or something. So do you think that using EVs, as a predictive or the tissue factors on EVs as predictive markers would be helpful, like at least for like people with high risk of developing this?
Yeah, it's possible. We didn't check, I have to be honest. The presence of tissue factor in healthy subject is really low in the extracellular vesicles. So healthy control that we use don't have an high expression of tissue factor, but we don't know if a person that has this such high reaction of a vaccine maybe had already a high level of tissue factor expression in their extracellular vesicles. So it could be interesting and worth to try to make some correlation and try to understand if these were already prone to develop thrombus formation, and if this could be already diagnosticated by extracellular vesicles. It's something that I really don't know, but it could be interesting to check for sure, and that could be really important for the clinic, I say. So it's really an interesting question, and it's something that should be investigated, in my opinion. Thank you.
Thanks.
So Vera asked if I can elaborate a little bit more on the MaxPlex. Sure, quite easy. Then I will send you also my email if you need. But the MaxPlex is really a super user-friendly kit, I have to say. So you have 37 fluorescent beads with antigen on the surface, with 37 different antigen on the surface. So these beads are able to bind your vesicles to the binding of the antigens. And then the day later, you can perform the detection with fluorescent antibody against CD9, CD63, and CD81 in order to only detect the extracellular vesicles that bind the antigen on the beads, because you can have also a piece of membrane with CD14 that binds the beads, but this piece of membrane is not going to be detected by the fluorescent antibody against CD9, CD63, and CD81. In this way, you have an analysis. You can evaluate how many CD9, CD63, and CD81 vesicles are also positive for the other 37 markers, and this can be detected by classical flow cytometry, because the beads are big and you can see them in the scatter and in the scatter plot and side scatter, and the beads are a little bit different in fluorescence. Each one is different in a little bit of fluorescence, so they can form 37 different small plot on your fax, and then you can discriminate which one is which antigen is which one. But yeah, I'm gonna send you also my email in the chat. If you want to contact me, I can give you more information. Any SARS-CoV-2 protein? No, we didn't check, or at least I have to be honest. We checked in the beginning, but we started really in the beginning, and antibody for spike protein that work properly weren't commercial at the beginning of the pandemic. Then we tried to see by electron microscopy if we can find something, and we found some structure that were similar to spike protein, but then we moved with we continued with this field of tissue factor, and we didn't check it anymore. But there is a paper, the one that I presented to you, that I showed you, that they performed proteomic analysis, and they found protein enriched in activation of immune response in mild and in severe. They found piece of a spike protein inside of the circulating extracellular vesicles. So yeah, there are papers that show that this vesicle can also carry spike protein, and this spike protein can have a role in the disease, so in the propagation of a disease. Can we then, after a V binding to the B stake, use a bis and list them? Yeah, sure. You can also use Maxwell's capture beads to capture your extracellular vesicles. We did it for sometimes for Western blot. You can also do it with RNA, but the problem is that you are with the Maxwell's capture beads, you are binding vesicles, or at least everything that has this antigen on the surface, so also piece of membrane of cells or other stuff that are positive for these markers. If you don't use then the detection with CD9, CD63, and CD81, you are not sure that you are analyzing extracellular vesicles. So if you use these beads to then isolate the vesicles and perform RNA analysis, you are not really sure that you are analyzing vesicles. If you want to do something with RNA, something specific for vesicles using beads, you can use beads binding to CD9, CD63, and CD81. There are a lot commercial type of this kind of beads that they are really easy to use and more specific for extracellular vesicles.
And then I hope you can capture enough to do any RNA analysis, because you always have too little, right?
Yeah. No.
Thank you for that, Carolina. Sorry, Madani. So you mentioned the other beads that would be better are which ones? Sorry, I didn't hear that.
There are different type of beads, commercial beads. For example, we use beads that are magnetic one and bind to CD9, CD63, and CD81 from SBI. I can also send you the, if you wrote me an email, I can send you the code of these beads. So are easy to use because you put these beads together with your sample. You can let them overnight to bind your vesicles, and the day later with a magnet you can isolate your beads and your beads with the vesicles.
Thank you very much. Thank you.
You're welcome. Okay.
Okay, I think all questions are answered now. So let's wrap it up. Thanks again also for the second half of this discussion. Thanks for the audience for interacting and coming up with your thoughts. Yeah, thanks for joining everybody, and have a good day, have a good evening wherever you are. Thanks a lot.
Thank you, everyone. Thank you, Tom. Bye.
Bye.
Thank you.